for some viruses. The nucleotide here, the structure of DNA, 3 component, the pentose sugar, 5 carbon sugar, DNA desoxyribose, and RNA ribose, compare the 2 carbon, and nitrogenous base, purine, adenine, guanine. This is the nitrogenous bases. Nitrogenous bases is 2 types, purine and pyrimidine. Purine, adenine, and guanine. Pyrimidine is cytosine, thymine, and uracil. Thymine in DNA and uracil in RNA. This is the structure of DNA, as we know. The ribose, the sugar, the 5 carbon sugar, and the nitrogenous bases. Phosphate group attached to the phosphate group attached to the 5 carbon. Nucleotides are linked by phosphodiester bond to form polynucleotide. Phosphodiester bond, covalent bond between the phosphate group attached to 5 carbon of the nucleotide and the 3 carbon of the sugar of another nucleotide. This bond is very strong, and for this reason DNA is remarkably stable. DNA can be boiled, even autoclaved, without degradation. The ends of the DNA or RNA chain are not the same. One end of the chain has 5 carbon and the other has 3 carbon. Here the 5 end are here the 3 end. Watson and Crick model, the double helix. Double helix model of DNA, 2 sources of information, base composition studies of Erwin Chergaev indicated double-strand DNA consists of 50% purine and 50% pyrimidine, and this amount of amount of T, Chergaev rules, and the amount of G equal to the amount of C. C percentage content varies from organism to organism. Here the Homo sapiens, the human being, A and T and G and C and G, C, this is the percentage of G and C and T. The mice and Drosophila, here we find Watson and Crick explain their model. The James Watson and Francis Crick in 1956 by the X-ray diffraction studies done by Rosalind Franklin and Merrick Wickens, conclusion DNA's helical structure. Here the picture taken by X-ray crystallography by Rosalind Franklin, and this is the first picture for the a bird biologist and expert on structure of charcoal, designer of underwater mine, a nuclear physicist. Those are the heroes of the story. The result, a nuclear physicist intersected, resulting not in the submarine explosion of feathers as one might expect, but in discovery that offered a glimpse of molecular mechanism that underlie all in the insight, innovation, and persistence of James Watson, Rosalind Franklin, Francis Crick, and Merrick Wickens led to detailed understanding of the structure of DNA, the stuff that genes are made of. There's all their knowledge integrated to discover the helical structure of the DNA. Double helix model of DNA, 6 main features. 2 polynucleotide chain wound in a right-handed clockwise double helix. Nucleotide chain are anti-parallel. 5 phase 3, 3 phase 5. Sugar phosphate backbone are on the outside of the double helix and the base are oriented toward the central axis. Complementary base pairs from opposite strand are bound together by weak hydrogen bonds. The pair with 2H bond and G pairs with the 3H bond. Base pair are 0.34 nanometer apart. One complete turn of helix require 3.4 nanometer 10 bases per turn. sugar phosphate backbone are not equally spaced, resulting in major and minor adenine thymine base double hydrogen bonds, guanine cytosine base triple hydrogen bonds, guanine cytosine triple hydrogen bonds, thymine adenine 2 hydrogen bonds. Here we find the double helix DNA. This is the backbone of the double helix, and those Nobel Prize in Physiology and Medicine, Maurice Buchan and Francis Crick and James Watson. Rosalind Franklin has a picture of the DNA by crystallography. RNA appear with U and C pairs with G. Messenger RNA, there is transfer RNA, ribosomal RNA, small nuclear RNA, all are single-stranded function in transcription, RNA processing and translation. Here we find the messenger RNA, its structure, the organization of DNA, RNA in chromosomes. The genome is composed of chromosome or set of chromosomes that contains all the DNA an organism or organelles possesses. The viral chromosome, single or double-stranded DNA or RNA, circular or linear, surrounded by protein. This is the mosaic virus, tobacco mosaic virus. This is the tutu bacteriophage. This is the lambda bacteriophage. Most contain one double-stranded circular DNA chromosome. Other consists of one or more chromosome and are either circular or linear, typically arranged in a dense clump in a region called the nucleoid. The problem measure linear. The Escherichia coli genome 4.6 MB would be 1000 times longer than the E. coli cell. How this genetic material find a space inside the cell? The human genome is 3.5 GB would be 2 to 3 meter long if stretched linearly. What is the solution? How the genetic material find the space inside the cell? The solutions are: number one, supercoiling. DNA double helix is twisted in space about its own axis. A process is controlled by topoisomerase enzymes. Occur in circular and linear DNA molecule. Looped domain. More about genome size. C volume. Total amount of DNA in the haploid 1N genome varied widely from species to species and show no relation to structure or organizational complex. Eukaryotic chromosome structure. Chromatin, complex of DNA and chromosomal protein. Twist as much protein as DNA. Two major types of protein, histone and non-histone. Histones, abundant basic protein with a positive charge that bind to DNA. 5 main types, H1, H2A, H2B, H3, H4, equal in mass to DNA, evolutionarily conserved, non-histone. All the other protein associated with DNA differ markedly in type and structure. Backing of DNA inside the chromosome into the chromosomes. Level 1, winding of DNA around the histone to create a nucleosome structure. Level 2, nucleosome connected by strand of linker DNA like beads on a string. Backing of nucleosome into 30 nanometer chromatin fiber. Formation of looped domain. Chromosome DNA is packed inside the macroscopic nuclei with the help of histone. These are positively charged proteins that strongly adhere to negatively charged DNA. And this is the level complex called nucleosome. Each nucleosome is composed of DNA around 1.65 times around each histone protein. This is the level DNA double helix. Beads on a string. Form of chromatin. Chromatin fiber of packed nucleosome. Extended section of chromosome and condensed section of chromosome. Here some terms about DNA. centromeric DNA, center of the chromosome specialized sequence function with the macrotubule and spindle apparatus during mitosis and meiosis. We need it in mitosis and meiosis. Telomeric DNA at extreme ends of the chromosome, main stability. It's responsible for the main stability and consists of tandem repeats, play a role in DNA replication and stability. And the aging of the cell. Unique sequence DNA, often referred to as a single copy and usually code for genes. Repetitive sequence DNA may be interspersed or clustered and vary